another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another fascinating episode of our show with another really interesting guest helping to create a better tomorrow on many different fronts. Today we have the honor of being joined by Dr. Charles Brenner, who is the Alfred E. Mann Family Foundation Chair in Diabetes and Cancer Metabolism, Professor and Chair of the Department of Diabetes and Cancer Metabolism at the City of Hope Comprehensive Cancer Center. With his Ph.D. in Cancer Biology from Stanford, Dr. Brenner's lab focuses on disturbances in nicotinamide adenine dinucleotide, or NAD, a central catalyst of metabolism in a wide range of diseases and conditions of metabolic stress. Among his most significant discoveries over the years, Dr. Brenner has identified nicotinamide riboside as a vitamin precursor of NAD, as well as a variety of quantitative metabolomic technologies that has allowed him and his lab to discover that the NAD system is disturbed by many diseases and conditions of metabolic stress, including diabetes and cancer. Dr. Brenner has also led the team that conducted the first clinical trials of nicotinamide riboside, establishing its oral bioavailability, participating in trials establishing safety and efficacy, potential activities of the compound in areas like clearance of liver fat and depression, of common markers of inflammation. His lab continues to research how the NAD system is dysregulated in a variety of conditions, including as a function of postpartum, coronavirus infections, various inflammatory conditions. The list goes on and on. Over the years, his work has been funded by numerous agencies, including the Leukemia and Lymphoma Society, the March of Dimes, Burr Wellcome's Fund, Beckman Foundation, Lung Cancer Research Foundation, as well as our NIH and National Science Foundations. Dr. Brenner's authored over 140 publications, was a senior editor of a book Oncogenomics, Molecular Approaches to Cancer. And when he's not doing all of that, Dr. Brenner also serves as a member of the Scientific Advisory Board and Chief Scientific Officer for Chromadex, a leading bioscience company focused on the domain
Scientific advisor. I'm a 100% employee of City of Hope, but I advise Chromadex, my own stock, and so forth.
Perfect. Which is heavily focused on healthy aging. All that being said, Dr. Charles Brenner, thanks so much for taking time out of your schedule to talk to us for a little while today.
It's my pleasure. Thank you so much, Ira.
You know, it's interesting. As I was going through your background, you know, that's really only a small part of it because you know, you have been involved in various forms of academia at Brandeis, here in Philly at Thomas Jefferson University, Dartmouth. But in the early days, you went into industry early on. You were hanging out at Chiron, DNAX.
Hanging out. That's what I was doing there. I was hanging out, right.
Talk a little bit about take us just a little bit back into the past.
Okay.
How you got interested in this whole area.
Okay, Bostonian, born in 1961, raised in New England, went to Wesleyan, graduated in 1983. It's kind of the dawn of the biotechnology industry. Yep. We didn't call it biotech then, it was called the genetic engineering industry. It was located sort of in two places, one of them in around Cambridge, Mass, which I knew well, and the other in the San Francisco Bay Area. So I decided, head west young man, graduated from college in 1983 with the quaint idea that you go to college not to be a burden on your parents. Nobody in my family had a doctoral degree, so I thought, you know, having a bachelor's in biology from Wesleyan qualified me to, you know, to get a job. And as you said, I worked at Chiron for a couple of years and a place called DNAX Research Institute, which was a wholly owned subsidiary of Schering-Plough already at the time. And there I learned, you know, quite a lot of technically about molecular biology, yeast genetics, and also about teamwork and what you could do with a group of fully formed PhDs that were working together, which is kind of not the way things are organized in academia, where you have one lab that does one thing and next to another lab that does an entirely different thing. So that was a long influence on me. But so 83, graduate from college. I've got 5 years of biotech under my belt by 88, and that's when I actually started grad school at Stanford. So I stayed in the Bay Area for about 10 years. First in 2 biotechs, then Stanford Biochemistry Department, where I got a Ph.D. in cancer biology, and I purified an enzyme that is involved in pro-hormone processing, very similar to the enzyme that makes insulin in humans, but we were doing the work in yeast. And then, so then it's 1993, got a PhD, and head back east, go to Brandeis for my postdoc, kind of a short postdoc. 96, I started running my own lab in 1996, and I did 7 years in Philly at Thomas Jefferson University. Then family and I moved to Dartmouth. I actually thought I was going to spend my whole career in the frozen north at Dartmouth. It's quite a great place. I had terrific grad students there. And as you know, we started really making the transition to NAD at Dartmouth, because that's where we discovered the nicotinamide riboside kinase pathway. And we did the crystal structure of nicotinamide riboside kinase and found other genes involved in NAD metabolism. We really found that there were a lot of things that people thought they knew that they hadn't really thought about very much, and realized that we could really make a big impact in NAD when I was at Dartmouth. And then didn't work out that I stayed there the whole time, because I got recruited to University of Iowa to be the head of biochemistry. So 2009, moved to Iowa City, further expanded my lab and program in NAD. And then last summer, 2020, City of Hope, made an offer I couldn't refuse, basically, and said, we're going to create a new department of diabetes and cancer metabolism. We think you're one of the few people that can do it. And so I moved my group out, and I'm now hiring faculty at City of Hope, to where everybody's interested in diseases of metabolic dysregulation, which include, cancer on the one hand, where it's always, pulling in resources to make a malignant clone and metastatic clone, and then diabetes, which is another constellation of diseases of resource misallocation. And of course, Ira, it all involves dysregulation of NAD, so it's a perfect place for us to be doing this stuff.
Excellent, excellent. And on your lab website, you talk about obviously NAD and the wide range of this is a coenzyme family. You refer to it as this crown jewels of metabolism, but they're not protected like the crown jewels in London. A lot of stuff. You're mentioning metabolic stress, these bioenergetic crises. Talk a little bit about what that means, because you list a couple different things. You talk about things that attack these enzymes, things that diminish them, and also at the gene level, things that alter their transcription and so forth. Talk about this basic basket of bioenergetic crises and what that means in terms of this very important set of enzymes.
Yeah, right. Okay, so the coenzymes, right? So NAD really refers to 4 related molecules. You spelled out the central one already: nicotinamide adenine dinucleotide, and that has 2 forms that either carry the electrons already, they're reduced to NADH, or they're in the form that's ready to accept electrons, which is called NAD+. And then NAD+ and NADH are in another form called NADP+ and NADPH. And these are the central catalysts of metabolism. So everything that our cell does, from turning food into energy, into ATP, to transmitting you know, signals down the length of an axon in a neuron, to flexing a muscle, to making stuff like making estrogen, androgen, making cell membrane, making more cells, repairing all of the molecules in the cell. All of those things are metabolism. And, you know, NAD coenzymes are important because they are the central catalyst of metabolism.
Mm-hmm.
And, you know, you repeated my new shtick, which is that you could describe these as the crown jewels of metabolism. They're so centrally important. But then it would surprise you to find out the crown jewels of metabolism are exposed to the conditions of metabolic stress. Like, so if in a storm of reactive oxygen species, NADPH, which is required to make almost everything in the cell,
Mm-hmm.
becomes utilized to basically detoxify free radicals. So reactive oxygen species disturbs NADPH. When DNA is damaged, which happens literally all the time, because oxygen can damage DNA, sunlight can damage DNA, inevitable conditions disturb DNA. NAD plus comes under attack. Enzymes called PARP1 and PARP2 convert NAD into a weird polymer called poly-ADP ribose. In coronavirus infection and virtually any other inflammatory condition, there's 5 or 6 enzymes related to PARP1, none of which actually polymerize ADP ribose, but all of which consume NAD. In the inflammatory conditions, these 5 or 6 other enzymes degrade NAD. Why? In order to try to protect the cell from an invader. And so the use case for ongoing NAD synthesis and potentially supplementation with something like nicotinamide riboside is that the NAD system comes under attack. And in frank diseases like heart failure, but also in the inevitable stresses of life, like going out into the sun, where, you know, solar energy will lead to pyrimidine dimers and single strand, you know, events that have to be repaired. So we think that NAD is a central protector of cellular resiliency and repair. We actually have a very specific set of things in mind when we think about resiliency. We don't think that resiliency is just like a psychological concept.
Right.
We think that resiliency has to do with cell survival and genome protection and resistance. Stress itself. Stress is not something that is the way you feel about something. Stress is a set of molecular events that disturb cell function. And homeostasis, which is maybe the most important concept in all of biology and biomedicine, homeostasis is how do our bodies try to repair themselves? How do we try to maintain the same temperature? How do we try to maintain the same roughly blood glucose? How do we desperately try to maintain the same weight over the course of our lives? How do we try to maintain our muscle mass? And it turns out that the NAD system is involved in all of those things. And because the NAD system comes under attack, the NAD system requires some support. And one of the things that we discovered, and people always ask me, what's special about nicotinamide riboside? Well, there's something special about all four of the ways that different cells can make NAD.
Mm-hmm.
Right. So some cells, like in the liver, some cells can make NAD from tryptophan, the amino acid. And in the human body, there's many cells that can make NAD from nicotinic acid, but not nerve cells, as it turns out, not neurons.
Okay.
All cells, from what we could tell, can make NAD from nicotinamide and from nicotinamide riboside, but when the NAD system comes under attack, we very frequently see nicotinamide riboside kinase genes get turned up, which
Okay.
basically says that the cell is interpreting a stress. It's saying that it needs more NAD, and it's turning up the NR kinase pathway. So the lucky mouse that was in the cages that Matthias Merricksay and I set up, this lucky mouse that was unfortunately unlucky because it has a genetic predisposition to heart failure, but the one that was given NR in its diet,
Mm-hmm.
it benefits because in the failing heart, it's turning up the NR pathway, right?
Mm-hmm.
And then if we provide that mouse with dietary NR, it can replete, it can fill up its NAD deficit in that failing heart because we've given it oral NR. But guess what it also means, Ira? And this is what, you know, our lab is focusing on. The fact that these genes turn up in conditions of metabolic stress
Mm-hmm.
means that there's an endogenous system that makes NR available to stressed tissues.
Mm-hmm.
And we don't entirely know what that is. We don't know where the NR, you know, is coming from. In the privacy of our group meeting, we have our ideas and, you know, we're trying to identify the endogenous sources of NR, but we also know that we can effectively boost NR availability orally in mice and in people.
It's fascinating. And, you know, thinking about, as you're saying, this theme of resiliency. I think no other place, I would assume, I'm not an expert on this today, but where you actually you don't have one, but you have two beings is in the postpartum situation. And you've recently done this work in postpartum conditions, showing all sorts of really fascinating things and supplementing the mothers with advancing the juvenile development or development advantage, weight loss. And once again, here we have two organisms that have major resiliency issues in postpartum. Talk a little bit about this. And this is very recent work you're just publishing, but whatever you can say about it, I really think it brings this concept.
Oh, it's published actually. So, you know, I was scratching my head, you know, 2-3 years ago, and I was thinking about, you know, the mouse heart failure experiments and other neuroprotective experiments where the most profound benefits of NR accrue to the kind of the sickest animals, right? Like in the heart failure model, there are mice that have a poor ejection fraction, right? They have a bad heart, and they have poor ejection fraction, and they have low fractional shortening, and then you give them NR, and then it basically gets corrected, right? You look at the healthy mice that don't have that genetic predisposition to heart failure, and the NR didn't do that much for them, right? And so why? Because they already had a perfect ejection fraction. You're measuring something that's already good, and it doesn't get any better, right? And I'm thinking, well, what a strange situation that I'm in, that I've discovered the vitamin activity of nicotinamide riboside, right? I found a company that wants, I founded the company.
Sure.
We identified a company with which to partner, a pre-existing company, Chromadex. that that wanted to commercialize this, right? Came up with a way to make tons of clean nicotinamide riboside chloride, put it through the review processes, new dietary ingredients, so forth and so on. And then it goes out to human beings, right? And it's blue bottle, and it says on the label, not intended to treat a disease or condition, right? And then I look at my mice, and I'm thinking the most profound effects are on the mice that have a disease or condition, right? Clearing fatty liver for a mouse that has fatty liver, but the mouse that doesn't have fatty liver, there's no improvement in fatty liver, right? So I'm thinking, well, you know, and I'm taking Niagen, and maybe my mile time is a little bit better, and I feel like I'm sleeping a little bit better, and I hear a lot of people say things like that. But boy, the opportunity is really with kind of diseases and conditions, right? And it's weird because it's kind of expensive as a vitamin, but it's sort of way too cheap to be a drug, right? Drug companies don't necessarily want to conduct a human interventional disease trial on something that someone can basically buy from Amazon or direct to consumer from Chromadex, right? Unless we were to come out with a much more powerful formulation, dosage, delivery system, and so forth and so on. So I'm trying to figure out how do I improve the human condition, with NR, if it's going to be more valuable to people with conditions of metabolic stress, but those are diseases, and companies aren't going to do those trials very readily. Then my 6-month pregnant assistant walked into my office right at that moment, and I realized, oh, that's good. Women are so amazing, right? And of course, we don't consider, pregnancy and we consider postpartum depression a sort of treatable disease or condition. But actually, every woman that has a baby experiences postpartum. Postpartum is the period in her life after she's given birth to a baby. Whether it has a full psychological manifestation or not, in her 9-month pregnancy, her body was transformed, right, to produce a whole nother human being. And in the postpartum period, in the immediate postpartum, her body is again transformed, where a whole different set of hormones come in order to mobilize protein, fat, and carbohydrate to her mammary to do the mammary biosynthetic program that produces the milk that is evolved to be the complete nutrition for the baby, right? Whether she chooses to do that a long time or not, whatever. That's the way that the mammals are built. And so suddenly I realized what I need to do is test pregnancy and postpartum as a metabolic stress and see whether the NAD system is disturbed, and we found it is. So most strikingly, at peak lactation, which is about 2 weeks, post-parturition, after the babies are born in a mouse or a rat, at peak lactation, mom's liver NAD system is dysregulated. She's distributing, not only is her liver distributing protein, fat, and carbohydrate to the mammary, which everybody knew that. Her adipose is distributing, her liver is distributing, a lot of stuff is distributing macronutrients to the mammary to make milk. But we discovered that her liver is also distributing NAD precursors to the mammary. And the mammary has a massively boosted NAD system. And that if you supplement mom with nicotinamide riboside to the point where her liver gets its NAD status back, to what it would be if she were not pregnant or postpartum. She gets her liver NAD status back. She actually makes more milk and higher quality milk, as a matter of fact. And she can have an advantage in weight loss, and the offspring have an advantage in neurodevelopment. And the offspring that are you know, on milk, on the offspring of NR supplemented mothers actually outperform the offspring of non-supplemented mothers into adulthood. So they have better balance, they have better motor skills, they have better spatial memory, they're less anxious. It's actually pretty remarkable. So, you know, we've done some follow-up experiments that we're going to be publishing fairly soon, and there's clinical trials now of NR in new moms that are registered at UC Davis. So we're really excited about this work.
Very exciting, really exciting. Let's go now in the other direction, because well, I see you sit at the City of Hope Comprehensive Cancer Center. I'm a couple, you know, down the street here from the Worcester Institute in Philadelphia, and as I'm sure you're aware, a couple of years ago there was this study focusing on cells, the senescent cells, showing that I don't know if it's a precursor compound to NAD had some issues with regard to inflammatory cytokines and so forth, and I know these are in vitro studies and stuff. I forget exactly the whole model, but once again, talk a little bit about this balance here, because obviously cancer is a condition where we're in extreme bioenergetic stress and everything that comes with cancer. I'm sure you remember this this Worcester study, but where are we with regard to sort of the optimization and when it comes to some of these cases where you know you don't want too much, but you don't want too little? Walk down the sort of the cancer path for us.
So cancer, I mean, NAD coenzymes are required for every cell in the body, you know, including, you know, immune cells that are trying to detect and destroy cancer cells. So in the largest trials that have been done of NAD precursors, they were nicotinamide trials in Australia. You know, daily vitamin B3 use was clearly associated with lower cancer incidence. And why? Because, you know, all of the things I already told you, that, you know, DNA, you know, repair processes require and churn NAD. And so higher NAD status is protective. There's multiple animal models that show that higher NAD status is protective. There are some, you know, studies that, you know, I think are relatively contrived, in which you have a particular cell line injected into a particular mouse, in which people have, you know, come to the conclusion that NAD is driving, you know, some sort of bad actor function in tumorigenesis. We found, actually, and this was a really nice collaboration with Ranjit Bindra at Yale, that in diffuse intrinsic pontine glioma, you know, which is terrible, inoperable brain cancer, the cells are malignant not because they have higher NAD, but they're malignant despite having lower NAD, and their NAD system is kind of rewired such that it's easier to kill them. We think that particular cancer genotypes are going to be targetable by, you know, inhibiting NAD metabolism. Disclosure, you know, Ranjit Bindra and I started a new company to try to, you know, develop those ideas and develop some compounds that will do that. But there's no evidence that NAD precursors would be contraindicated for, you know, for people that are concerned about cancer incidence. Like I said, the largest clinical studies in Australia indicated that NAD precursors are likely cancer protective. And then importantly, we're very interested in neuropathy. We showed that oral NR protects against development of diabetic neuropathy.
Yep.
In the mouse, and then in collaboration with Donna Hammond at the University of Iowa, we showed that oral NR can protect against and potentially reverse chemotherapeutic neuropathy. And then Donna went ahead and registered and is conducting a human clinical trial in breast cancer patients where she wants to see whether oral NR will be protective enough against the neuropathic effects of breast cancer chemotherapy, that women can stay on drug if they're supplemented with NR. And then everyone asked Donna Hammond to test whether NR was pro-carcinogenic in her rat model, and she has found that it's not. She's published that. So we don't think that there's really a concern about higher NAD status being carcinogenic.
Excellent. Excellent. Let's go to the other. You wear the cancer metabolism hat, but you also wear the diabetes hat. You Alfred E. Mann Family Foundation chair. It is a legend in the biotech space on many fronts. Obviously, diabetes was clearly one of the things he was heavily focused on. You mentioned the type 2 diabetes and neuropathy work, but also, you had a fascinating paper that focused on the microbiome and applications potentially there. Say a few words, if you would, about sort of the connection. Once again, the microbiome, part of us, but separate organism in a sense. But some of the interesting things you're finding out there, just because the microbiome is such a hot topic.
Good question. So City of Hope, by the way, is not only a comprehensive cancer center.
Sure.
It's also, probably 65% or so of the research at City of Hope is cancer, and another 35% of it is diabetes and metabolism. And the unit, my academic department, is actually part of the Arthur Riggs Diabetes and Metabolism Research Institute.
Yep.
So I'm a member of the Cancer Center and a program co-director, as well as a department chair in the Diabetes and Metabolism Research Institute, and we're interested in both, right?
Yep.
So, so microbiome, microbiome is fascinating. I don't think it's really actionable yet. I'm not motivated to sequence my own microbiome or anything like that. There's a lot of things that people are wanting to self-test that I think are, great business plans for people selling those tests, but don't actually provide any added value to the person. But clearly, there are profound interactions between the microbiome and our metabolic health. There are not fully understood connections between the microbiome and NAD metabolism. One thing that we do know real well is that all cells in the body will convert NAD to nicotinamide, and many cells will use nicotinic acid, and really the only known enzyme that will convert nicotinamide to nicotinic acid is a bacterial enzyme. So we know that there's a relationship between our microbiome and the availability of nicotinic acid, at least in some tissues, like in the gut. There may be other tissues where there are other ways to make nicotinic acid, but when I draw a diagram of NAD metabolism, I definitely include the microbiome in one of the arrows. But it's a profoundly interesting area, and some of our digestion is carried out by our microbial friends. Fermentation is something that that has been with human civilization forever, and it's a growth area in science.
Thank you for that. Now I'm going to say a dirty word, aging. You early on, you were the first person to show the connections between
Yep.
a lot of this NAD work and sirtuins. You've not
Yep.
gone, your work has gone in many other directions. Aside from your work at Chromadex, you have in essence spawned certain industries indirectly, whether you wanted to or not. But take the floor for a couple minutes and just talk about
Okay.
aging, longevity.
Aging.
If you want to go into the current social media thing, you can or not,
Okay.
but
All
please
right.
have the floor.
Yeah. Okay. So aging is really important, right? So we're all aging, and aging is, in part modifiable. It's very modifiable, I think, by physical activity, diet, clean water, vaccination. I would argue not being in a household with guns. There's a lot of things that, actually family status. I think that men that are married and people that are coupled, you know, men demonstrably live longer, when they're married, but I'm sure that probably bad marriages are probably worse. And so there's a lot of things that we can do that can allow us to age better. When Rob Fried and I sat down and talked about having a direct to consumer Niagen, we agreed to put age better on the bottle. But I don't like the term anti-aging.
Mm-hmm.
I don't like the term anti-aging because to me, the term anti-aging is too associated with hype and false claims and false promises that have been made for literally thousands of years.
Mm-hmm.
About fountains of youth and potions that would reverse aging or make aging optional. And I don't think that aging is well enough understood. First of all, I think that anybody that claims otherwise is not being truthful. And I just don't want to associate my own science with that term. Now, in 2007, which was 3 years after we discovered the nicotinamide riboside kinase pathway, we did an experiment in yeast. And we said, here's SIR2 dependent aging in yeast,
Mm-hmm.
and we can extend lifespan. It looks like it's increasing SIR2 activity using nicotinamide riboside, and we published that, and no one's ever challenged that paper. It's a solid paper, but I didn't extrapolate from that. I didn't extrapolate from that paper that because we could extend lifespan in yeast, and because we can activate SIR2 in yeast, that we could extend lifespan in humans. And since that time, it's become eminently clear that the role of that SIR2 is a legit anti-aging, and I will defend SIR2 as an anti-aging gene in one model of one yeast.
Okay.
Literally in one model of one yeast. So SIR2 deletion mutants live longer in the other model, which no one ever talks about, but Walter Longo showed that SIR2 mutants live longer.
Mm.
So that makes SIR2 both pro-aging and anti-aging in different models in one yeast. But then SIR2 was claimed to be anti-aging in worms and flies, and it's not. That was basically debunked. And the stuff in mice is just hopelessly contaminated with people's expectations, right? So when you have, dozens and hundreds of papers saying that SIR2 and SIR2 related genes are anti-aging, then you're setting up expectations in a laboratory and the people that are doing experiments to see, let's see if we can demonstrate it's anti-aging in flies and worms. People did that, and it turns out it's not true, right? They didn't actually, in science, you make more progress when you test your hypotheses. You try to disprove your hypotheses, rather than saying, oh, let's assemble more evidence in favor of our favorite idea, right? And then it becomes very easy for people to say, here's more evidence that SIR2 or SIR2 related genes do all of these wonderful things. And I've described it as scientific cultism, and it really is, because if the underlying premises have been disproven, and then people are still trying to find evidence that it's doing something, and then they don't find that SIRT1 does something, then they say, oh, it must be some other sirtuin related gene that does that. It's actually ridiculous. It's like saying, you know, you think that I don't have a brother, but it's like saying, you think that I committed some crime, right? And then you do a test to see if I did it, and then I didn't do it. And then you go and you look at 6 of my brothers, and then and then you find one of them was in some way related to it, and then you say, see, we were right. So there's some real nonsense out there. And then resveratrol is actually worse, because resveratrol, you know, is a well-known compound that, you know, is in wine. It's also in peanuts, by the way. And it's an antioxidant, and it binds to a number of different proteins in the cells and may be associated with various things. But there was this idea, you know, that based upon experiments that cannot be reproduced, that resveratrol is an activator of human SIRT1, and resveratrol extends lifespan in yeast, but resveratrol doesn't extend lifespan in yeast, and resveratrol doesn't interact with SIRT1. And now the people that say that it does activate SIRT1, they are saying it activates SIRT1 in a way in which they couldn't have discovered it the way they said they discovered it in yeast. So the whole thing is really, really messed up. And it actually is, it's not harmless, right? Because it leads companies to invest in programs that are not well premised, that are going to fail and lead to massive financial losses and layoffs, which has absolutely happened. It leads to people taking dietary supplements that are not evidence-based because someone charismatic tells them something, and it leads to an atmosphere that is not scientific, where you know, people can basically claim things on the basis of their charisma and not accept falsifiability. And so I really have a problem right now with some people's interpretation of the value of NAD biology. Because the value of NAD biology is based upon 4 NAD coenzymes being the central catalyst of metabolism that get disturbed in, you know, many, many conditions of metabolic stress, and NAD coenzymes being required for hundreds of different processes, right? And then it's a very naive and inaccurate view that the value of NAD is to increase the activity of SIRT1, and by doing so, it's going to have anti-aging properties. I don't like that at all. I think it's not true. I think that NAD it's just misleading.
Mhm.
I do think that higher NAD status provides cellular and tissue resiliency. I do support clinical trials as well as all kinds of preclinical research that is determining the efficacy and mechanisms of action of nicotinamide riboside. But I think that the field has been distorted by, by some views that are not evidence-based and that it is harmful.
I appreciate your insight on that matter. Charles, while I have you, obviously you have a broad range of programs at the City of Hope. The lab is hiring. It's on your website. Any other stuff that you want to mention? Upcoming conferences, talks you're going to be giving? Anything else you want to talk about while I have you? I'll give you the floor.
Okay, well, that's great. I really appreciate it. So things are, opening up again. I don't have my calendar right off the bat, but I know I've been invited to give in-person talks at Yale and UC Davis. And I'm hearing things, all the time. I mean, the great thing about working on NAD is that, my phone's always ringing and emails always buzzing with people that are running into the NAD system where it's dysregulated, and rare diseases and common diseases and neurodegenerative, conditions in which, we could potentially make a contribution. But I just want people to stay evidence-based, not get over their skis. The way that we, introduced Niagen was, I think, putting safety first and, going through a process in which, a clean manufacturing process was developed, and materials were reviewed for new dietary indication and generally regarded as safe. And human clinical trials have been done that have always emphasized safety. And now with 45, 50 clinical trials registered for many diseases and conditions, we think that there are going to be exciting things coming out. I mean, just in the last month, there was a study that came out that was peer-reviewed from Turkey and Sweden, in which nicotinamide riboside, high dose, so like 2 grams a day, was included in a cocktail of essentially over-the-counter molecules like N-acetylcysteine, serine, and nicotinamide riboside, N-acetylcysteine, serine, and L-carnitine, I believe.
Okay.
And I don't fully understand why the other 3 were included, but the trial was for COVID recovery. And we published a paper last year on how coronavirus infection in cell systems attacks the NAD system. We understand how that works.
Yep.
And we made predictions and had some pretty convincing preclinical data that a higher NAD status would boost the innate immune system. And we were very encouraged to see that this dietary supplement mixture that included high dose NR greatly accelerated time to recovery of mild to moderate COVID patients, placebo-controlled studies. So we think that there's some exciting things coming out, but we want to see things done safely, and we just don't want people to think that it's magic or miraculous. We think you can age better. I'm 59, and want to stay active for a long time, but at the same time, I'm excited, and I think there's a lot more stuff that's going to be coming out in the years to come.
It's exciting times, and we'll be following you and reading you on, Charles. Really great.
Great. So people can ask me questions on Twitter at Charles M. Brenner, and look at our website, which is brennerlab.net. And thanks for having me, Ira.
Surely, we'll put all those links in the
Okay.
bio as well. But to wrap up, for everybody that's going to be listening to this episode on the podcast or watching on the YouTube channel, you've been listening to Dr. Charles Brenner, Alfred E. Mann Family Foundation Chair, Diabetes and Cancer Metabolism, Professor and Chair in the Department, City of Hope Comprehensive Cancer Center. Charles, once again, thanks for taking the time out of your schedule to talk to us. Thanks for everything you're involved in. And as we say on our show, thanks for helping to create a better tomorrow through all your work. Really very inspiring.
Thank you.